So the last of the historical figures in microbiology is Alexander Fleming. Fleming is responsible for multiple discoveries in micro, one being he discovered the enzyme lysozyme. So this enzyme is commonly found in our tears, sweat, and saliva, and it is an enzyme that can kill bacteria by breaking down their cell walls and causing them to burst through cell lysis. So he did have that discovery. What he's most famous for, though, is discovering the first antibiotic, which was the antibiotic that we know as penicillin. It was by complete accident that he discovered this. He was essentially working in his lab and was growing a culture of bacteria. And what he did is he actually left the lid off of the culture of bacteria for a weekend. And we typically never leave the lids off of petri dishes because what can happen is airborne contaminants can get in and contaminate our culture. But that's what happened to him. When he went back on Monday, he noticed that there was mold growing all over his petri dish that he had used to grow bacteria. So when he was going to throw it away, he questioned, there's all this mold here. Where did the bacteria go, though? The bacteria that was in there previously is now gone. And what he basically concluded was that the mold that was growing in the petri dish must have produced some type of chemical that was able to kill the bacteria. Now, we now know that mold was the penicillium mold that produces a chemical that we know as penicillin that is able to kill bacteria. But at this time, there was no antibiotics present. So if anyone got a bacterial infection as simple as a cut, it could lead to death or amputation. So him discovering this chemical that was able to kill bacteria was life-saving for many individuals and kind of started off the use of antibiotics in regards to treating bacterial infections. So he's most famous for discovering penicillin as an antibiotic. A little bit about microbiology today. We'll kind of utilize microbiology throughout the course of the semester. We'll be talking about all different types of things. But one thing we will focus on throughout the lesson today and then later on are bacteriophages or bacteriophages. These are viruses that are able to infect and kill bacteria. We have one pictured here. I know it looks like a crazy alien, but this is how they naturally look. And they can be made in a lab, but they can also just exist naturally out in the soil. So they do look like this, where they have these little heads that has their DNA stored in it, and then they have these little necks, and then these little legs that they use to bind to bacterial cells. But essentially what they do is they bind to a bacterial cell, they inject their DNA into the bacterial cell. That's them infecting the bacterial cell. And then inside of that bacterial cell, they replicate until there's millions of them, and then the bacterial cell bursts and dies and releases all the viruses, which then go infect new bacteria, and the whole cycle just continues. So what we're really doing is we're killing bacteria. These are viruses that can infect and kill bacteria, so we tend to kind of think of them as a good thing. So who cares? Who cares about viruses that can infect and kill bacteria? I guess we tend to think of viruses as something bad, but there are good viruses. So the reason why we care so much about bacteriophages is because right now what we're using to treat bacterial infections and kill bacteria are antibiotics. And I think we all know that bacteria have become resistant to antibiotics. So it's not unreasonable to assume that possibly in our lifetime there may be many antibiotics that are non-effective anymore. So what are we going to do when we get to the point where antibiotics aren't able to kill bacteria like they used to be able to? We need to have some other type of medication available when we get to that point. And that could potentially be bacteriophages. They can kind of supplement antibiotics. So if a bacterium is resistant to all known antibiotics, potentially a patient could take a capsule of bacteriophages that would then go into their body and infect the bacteria and ultimately destroy the bacteria. So kind of combating antibiotic resistance is one way that bacteriophages could be beneficial. Another thing is that bacteriophages are very specific, whereas antibiotics can sometimes have a broad spectrum of activity. So when you take an antibiotic, it's meant to kill bacteria, which is good. It gets rid of your infection. But it can also kill some of your good bacteria, which we call the normal biota or the normal flora. And that can cause side effects. If you've ever had an upset stomach or a yeast infection after taking an antibiotic, what you're experiencing is the fact that the antibiotic harmed your good bacteria while it was trying to get rid of the bad bacteria that was causing the infection. So that's kind of one of the downsides of taking antibiotics, is they can kill the good and the bad bacteria. With bacteriophages, they're highly specific. So they can go in with needle-like precision, kind of like that magic bullet theory, and get rid of just the bacteria that's causing the infection and leave all of the good microorganisms, your normal biota, leave those alone. So in that sense, bacteriophages could be very beneficial. As far as how they're being used today, they're not officially in use as of right now. They're not FDA approved. The problem is that while many bacteriophages seem to be beneficial and would help us cure infections, there is kind of a dark side to it that we'll talk about in a later lesson, where sometimes a bacteriophage can actually infect a bacteria, and instead of killing it, it can make it stronger. So obviously that would not be ideal. So there's still a lot of research being done in regards to bacteriophages and how we can utilize them, but there are some obvious risks. One beneficial way that we could potentially use them one day, other than to treat bacterial infections, is to protect against foodborne contaminants and things like food poisoning. One bacterium that causes fairly severe food poisoning is the Listeria bacteria. This bacteria can be found in food, specifically food that requires refrigeration, and it can cause a fairly severe form of food poisoning with vomiting and diarrhea. Bluebell ice cream, years ago, their facility was contaminated with Listeria and they had to shut down. This bacterium is the reason why they recommend that pregnant women do not eat lunch meat or deli meat, because there is the potential for this bacterium to be present and it could be harmful at making pregnant women sick and leading to dehydration. So one potential use of bacteriophages are using them in packaged foods. So what they could do is they could package foods with the bacteriophages. And that way, if the food gets contaminated with the bacteriophage could infect and kill the bacteria. So that way, when you ingested the once contaminated food, the bacteria would already be dead and long gone, and it would not make you sick. So there is potential one day that these bacteriophages could be packaged with food, almost like a preservative, to prevent against foodborne illness. And yet another thing, we kind of already discussed this at the beginning, another big way that microbiology is being used today is in this field of recombinant DNA technology, which is a type of microbial genetics where we can basically program bacteria to produce human protein, because there are different diseases where humans are not able to produce a certain protein for themselves, so they have to take supplement forms of that protein, maybe through a pill or an injection. But where do those proteins come from so that they can take that? Well, they can be made by bacteria. So like I said, this is often done with insulin. It used to be that to get insulin, animals had to be slaughtered, and then that would allow us to have the insulin that we could use for injections, but it's hard to keep up with that amount of insulin and the growing rate of type 2 diabetes. And then also, because it wasn't human insulin, it was animal insulin, some individuals were having allergic type reactions to it. So this is definitely more sustainable. What they do is they just take the recipe for how to make insulin, which is in our DNA, it's in the insulin gene, and they basically put that into a bacterial cell. And that manipulates the bacterial cell into producing the protein that we know as insulin. They don't really know why they're doing it, but they have the recipe to do it, so they do it, and then we extract it from them, and then we use it medically to fight diabetes. So we'll talk in more detail about recombinant DNA technology once we get to the microbial genetics lesson.